The overall goal of this procedure is to generate and characterize T lymphocytes from induced pluripotent stem or iPS cells. The iPS cells can then be differentiated in vitro on an OP9DL1 culture system and then matured in vivo in a RAG-deficient mouse. Or the cells can be genetically engineered to overexpress OT1 TCR and then adoptively transferred for in vivo development. After 6 weeks of in vivo differentiation, the mice can be challenged by intraperitoneal injection with EG7 tumor cells, and then the antigen specificity of the iPS-derived T cells can be evaluated. Ultimately, the iPS cells can differentiate into both conventional and antigen-specific T cells, the latter of which are able to protect animals from tumor challenge.
The implications of this technique extend toward individualized cancer immunotherapy. The method allows the generation of large numbers of tumor-reactive T cells derived from minimal amounts of patient's blood or skin tissue.
On day 0, transfer 5 × 10⁴ iPS cells onto a 100 mm culture dish containing a confluent OP9DL1 cell monolayer in 20% FBS alpha MEM media. On day 5, trypsinize then centrifuge the cells. After incubating them on a fresh 100 mm culture dish for 30 minutes in 37-degree incubator, plate 5 × 10⁵ of the floating cells in 20% FBS alpha MEM media and mouse FLT3 ligand on a new 100 mm dish containing a monolayer of OP9DL1 cells. On day 8, gently pipette down the loosely attached cells